Hello, everyone who is trickling in. We're going to get started here of our 35th installment of the Phenotyp Speaker Series. We are so excited to see all of you joining live here. We do love that this is a live show that is later produced into a podcast, which is really exciting. We get to do both. For those that don't know, I'm your host, Kira Dineen, and today, as you can see, we're going to be talking about matchmaking for rare disease diagnosis, which is very apropos as Rare Disease Day 2026 is coming up in just a few days. For those that aren't aware, the reason we have Rare Disease Day and month in February is because Leap Day is sometimes in this month. So when there is February 29th, that is Rare Disease Day, because it's the rarest day of the year. I remember when I learned that and being like, oh, that totally makes sense. So when the 29th is not there, it's the last day in February. So yeah, for everybody joining in, we're going to be talking about matchmaking for rare disease diagnosis. And we really appreciate you joining in, whether it is your lunchtime, dinner time, breakfast, wherever you are in the world. For those that might be new to our series, the way this works is I get to interview leaders in the field about the topic we're covering. And after I ask my questions, we get to the exciting part where we ask your questions. So make sure you're using the Q&A box if you are watching this live, because I want to make sure we get to your questions. But don't wait until the end because you might forget your question. So as you think of them, use that Q&A box in the Zoom here. And Phenotyps is the lovely sponsor and organizer of this amazing series. And many of us know that it is a complete EHR integrated solution for medical genetics and has created software and services that ease genetic professionals' workflow and improve efficiency. Phenotyp's genomic health record includes tools like inclusive pedigree builders, automated pedigrees, human phenotype ontology capture, built-in cancer risk assessment, diagnostic insights, and so much more. We have all experienced, I bet, at some point, if you've ever been in clinic or a clinic associated position, electronic health records are not built for genetics. We're kind of siloed. It's some tab that's buried somewhere. It is not set up in a way that is easy to use. It's clunky, which is really what inspired Phenotyps to be able to develop the genomic health record, because it fills in those gaps to provide a unified and seamless genetics workflow for all of us. And it's designed to capture this genetic data in machine optimized formats that propel rare disease diagnosis by making it easier to securely collaborate, run algorithms, AI, harness matchmaking, which is what we're going to be talking about today. So as I mentioned, I'm Kira Dineen. I'm the host for this webinar series. I'm also the CEO of Genepool Media, which houses a bunch of genetics and science related podcasts, including this show, Phenotyp Speaker Series, and our flagship show that I'm the host of, DNA Today. I'm also a certified genetic counselor. But enough about me and what we are going to talk about. Let's just dive into it, because I see that we already have a lot of people that have joined us live. So I'm going to have our three amazing panelists introduce themselves. They are strategists, developers, and researchers behind patient matchmaking initiatives. Orion, I think I'm going to have you start us out with introducing yourself.
Sure. Thanks, Kira. Nice to be here. My name is Orion Buske. I'm the CEO and co-founder at Phenotyps. My background is in computer science. My PhD was spent working on algorithms to help diagnose people with rare genetic conditions and working on the Matchmaker Exchange in the early days before we spun Phenotyps out. So really happy to be here and joined by you and Nara and Matt. Have a little bit of a reunion and get to talk about this topic that's really dear and true to my heart.
Yeah, you're the perfect person coming directly from Phenotyps to be on the show, because like your background is basically the synopsis of what we're going to be talking about. So thank you so much for being able to join. And Matt, do you want to introduce yourself next?
Sure. So thank you again, Kira, for having me on the show. As mentioned, my name is Matt Osmond. I'm also a certified genetic counselor, and I've been working as a project manager for the Care for Canada Consortium for the last 8 years, based out of Ottawa, Canada. And so I've really worked at the intersection of genetics and computer science on a number of things related to exome and genome data analysis, rare disease matchmaking, as we're going to talk about today, as well as sort of larger data infrastructure projects across the country. So thanks again for having me.
Yeah, wonderful. And love to have another genetic counselor on the call to be able to represent in that way. And again, your background is just so perfect for this. And Nara, can you introduce yourself and what you're bringing to the table?
I'm Nara Sobreira, and thank you for having me here today to participate in the program with Orion, Matt, old friends that I have been working with for a long time. I'm a clinical geneticist and I do research on rare diseases using next generation sequencing and other kinds of types of omics to find the genes that cause the rare disease that we don't understand the genetics of them. And as part of that, I have been developing tools to analyze the data, but also to share the data. And that's how I started participating on these efforts. Now, I think 15 years ago, when we launched the Matchmaker Exchange. Um, so happy to be here and talk about it as we continue working on these projects and trying to make data sharing better for everybody.
And with you being with these initiatives and doing this research for so many years, hopefully we'll be able to get some of that historical context of just where we've come from, where we are today, where we're going in terms of this matchmaking for rare diseases. And I figure we should probably start with a little bit of background on just defining like what is patient matchmaking before we start getting into the weeds with everything. Nara, I would love for you to just continue explaining that and giving a little bit more background for people that are not familiar with this term and maybe said, hey, I want to check this out because I don't know what it is.
Yeah. And actually, we don't use the term patient matching. We actually use the gene matching term and now the variant matching term because we actually decided, or it was easier for us to start by the gene when we, in Gene Matcher, that's the tool that I started working with 15 years ago. And Phenome Centro that came before Phenotypes also started at the same time, and they were trying to match patients by the phenotype, and we were trying to match patients by the gene. But at the end, we were trying to do the same thing, that is finding patients with rare disease. That's a very difficult thing when we are talking about patients that are only 8 patients with that disease in the world. How do you do that? Meanwhile, 15 years ago, it's when we started using exomes and genomes, and that was creating a lot of candidate genes per patient. And to understand what was the best candidate, we needed to find multiple patients with similar phenotype and mutation in the same gene. So there you understand why Phenome Center at the time said, I want to look for the patients based on the features. And we said, let's try to find the patients based on the gene because these are the two main pieces. The patients have to be the same phenotype and have mutation in the same gene. So we came in different ways, but they overlapped. And that's when we decided that many tools would probably be coming up doing this type of work, and it didn't make sense to us to have the users, and the users being anybody who is trying to solve one case or understand the cause of that one case and find the diagnosis of that one disease for that patient. Didn't make sense to them to go to 10 different databases. I have to look here, I have to look here, I have to look here. So that's when Matchmaker Exchange was created, because they said, why don't we create an API that will connect all the databases that are doing this similar type of work? And at the time, Gene Matcher and Phenome Central groups, teams got together and started developing the API that connected first Gene Matcher to Phenome Central. And that continued growing. The network continued growing. And now we have more than 10 databases around the world that are connected through that API. So that's how we started. I'll let others chime in as they also joined when we started this work. But it's how it started. Not to say that that was the first that it had happened. Data sharing was happening before. For example, Decipher, that was the third one to join. Decipher was already up and running and sharing data of patients with rare diseases based on copy number variations, another type of variants. But I don't think that databases were connected. That was the first time that two databases were actually connected, and then others started joining. So that's, I think, the innovation at the time was connecting these databases.
The data itself, I think, is so important that you're providing because years ago, the most data we probably had was the appearances and different the phenotype aspect, and now we have so much more rich and useful genetic data, so it makes sense to pull these both in. Orion, further that, because obviously I'm very much like understanding this from a surface level. Am I on the right track in terms of like the data really driving how we're able to use this type of matchmaking?
Yeah, I think certainly as sequencing has gotten more affordable, more mainstream, we're doing the sequencing sooner, faster. That helps with the systems then having that kind of be part of the core for that matching. I think also it was just these were research projects and innovations that were being developed early on, and we were trying to figure out what the best approach was. I think Phenome Central was taking more of a phenotype first approach to matching. Gene Matcher was taking more of a gene first approach to matching. I think it Gene Matcher's approach ended up ultimately being kind of the right one. And so then you see the other systems starting to then adopt that and incorporate that. And that's how overall we learn. I think we were, starting out with a bit of a friendly competition with our different competing tools. And then we quickly realized that it would all be stronger and more effective for sort of solving the core problem by working together and federating these systems. I think the core challenge is that rare diseases are rare, and so really all of these were about trying to break down silos for data sharing because that is how you find these additional unrelated families that are necessary in order to have a sufficient cohort to be able to ultimately turn a gene of unknown significance that might be a candidate into something that you can publish. And now as part of our knowledge about, here's another disease gene, we can now diagnose people reliably who have conditions, who have variants in that gene, and kind of expand the knowledge and our ability there. And so that means that each matchmaker that was being developed at the time, or matchmaking database, was trying to solve that in their own areas, like connecting different institutions that otherwise might not have mechanisms for sharing data of this sort in order to find those additional families, and then ultimately through the matchmaker exchange connecting all these databases around the world.
And Matt, tell me with your perspective coming from the genetic counseling angle, other thoughts to add on?
Yeah, I think both Nara and Orion have really summarized well what historically was the impetus for starting a network that was dedicated to matchmaking. And I think even beyond the idea of looking in a single database, I know from speaking to other geneticists, there were other methods that people tried to use to find similar undiagnosed rare disease families out there in the world. And it was really about like who you knew in the scientific academic communities. You would go to various conferences all over the world, and you'd sit there and you'd listen to a bunch of abstracts being presented of patients who were unsolved, and you'd go up and you'd talk to someone after the meeting to try and connect. There's a famous story from the main PI for Care for Error, Dr. Kim Boycott, saying that while she was in transit, she just so happened to pull up a paper on her phone that happened to be a case description for a patient who had the exact same gene of uncertain significance, and that's how she matched with someone. And so, we had these, you either looked in a single database, or it was really about, whatever connections you had in the scientific community. And And the Matchmaker Exchange and the variant level matching network are really trying to, make those connections much easier, as mentioned, because we have this exome and genome data that can generate these genes of uncertain significance. So finding changes in genes that we don't yet know whether they're associated with human disease or not, like because those opportunities are coming up more and more often with this data, like having these networks in place is really important for trying to get those answers to those families.
Yeah, yeah, definitely. And I think that begs the question, like, who is actually using these matchmaking platforms and how they use them? Because I'm thinking like, oh, genetic counselors, geneticists, but it's beyond that. It's hopefully not only people that are working professionally in our clinic genetics field. Does anyone want to provide a little more context of, hopefully who's using them and how they are? Nara.
I can say something because I think it's a very important thing that we have aimed from the beginning, that is actually get it out of the research setting. Like Matt was just saying, Kim happens to be a clinician, a researcher who has access to these papers and could actually read the paper and think that that patient had the same phenotype that her patient had and mutation the same gene. But not everybody has access to papers, and not even researchers in some countries have access to papers easily. In Brazil, for example, where I did most of my training, we didn't have easily accessible papers, right, to be reading as they came out. So what these tools did and are doing, it's actually making that network available for people who don't have access to papers, who are not going to the meetings in other countries to be going through the abstracts that are being presented, who really don't know everybody, to be just sending a message by email or calling and saying, do you have a patient with mutation in this gene or with that one mutation or this one phenotype? So these networks are improving that accessibility. Everybody in every country now can just access one of these tools online and query all these other databases that are connected in the network with one single step. So that open up the network and to the users everywhere around the world. First thing. Second, it also took out of the researchers' hands. Now other people can actually take the step to look for these other patients and find the answers and the diagnosis. It can be the clinician, it can be the counselor, it can be any healthcare provider that is interested on that case, on that patient, and that diagnosis. And it can be the patients themselves, right? So we have patients that we know participate in the network in our tools like Gene Matcher and Variant Matcher. We know how we ask them to identify themselves as we ask everybody else to identify like what is their roles. Are you a researcher? Are you a clinician? Are you a healthcare provider? Are you a patient? So we actually know who we are serving and how to best serve them. But we have patients now all around the world who just get that report and see that there is a gene that's a candidate, and they will go after all the patients themselves. And the tools help them to have this access, what without these tools would be impossible for them. So we have in GeneMatcher researchers, clinical geneticists, counselors, healthcare providers in general. The researchers are not only human genetics researchers, but we have model organisms researchers, and we have patients using. And now we want to expand it, that not only research data is being queried and participating in the network, but also data that is being created by clinical laboratories and data that is being stored in hospitals and data that is being stored in patient-led databases. that they can also join the network and with that make the searchable data much richer and help us to find solutions and answers much faster than what we are doing now.
Yeah, the more it's built up, the better of a chance there are for these matches to occur. We need to keep growing this data. And I'm thinking in terms of like we've learned about like what this concept is, why it was important for these databases to be built and the tools around them so that all of these different healthcare providers, patients, caregivers can take advantage and really be using this as part of the diagnostic odyssey to end that sooner in rare disease. But how does the technology behind the platform really work? If we dive into the details a little bit more, how does it identify the matches in terms of like what that workflow looks like? Orion,
I'm
yeah, I would
happy
love
to
to
answer
learn
a
about
little bit of
this.
that, but also hand it, Matt, I know you've been working on how the sausage is made for a very long time. So happy for, I'll keep maybe my response very short. But at the end of the day, I think a lot of what these systems have been built on top of is having structured data for the phenotypic information and structured data for the genetic information. By structured, we mean not usually not just words, they're mapped to some terminology or an ontology that allows a computer algorithm to reason about them and calculate similarity using some method. So the most common one for the phenotypic side, for the clinical presentation, is the human phenotype ontology. And then for genetic information, having a gene ID or gene symbol to match across is really important. And that allows a lot of these algorithms to find direct matches for gene and then cluster or describe or calculate phenotypic similarity and do clustering on that side, which is something that we have feel is very important for continuing to help with that review and analysis by the people who end up with matches. But Matt, maybe you can talk more about that since that's a huge part of what you've been involved in.
Yeah, of course. So I agree with you, Orion, in terms of the fundamentals of what allows matching to happen. The key part of it, especially when you have databases that have data structured or stored in slightly different ways, is you have to use similar standards to one another so your systems can communicate back and forth. And that's a really great starting point for finding potential matches within your database and across to other databases. I think the other part that's really important to mention to those who might not be familiar with the idea of matchmaking and the matchmaker exchange is we've been talking a lot about computer systems, but there's still a big human component to matchmaking as well. So, finding out that there is another patient in another database separate from yours where they have a variant, a change in the same gene that you're really interested in is a great start. And you may have some level, if you're lucky, some phenotype on both sides, some clinical features that you say these could be potential fits for one another. But at the end of the day, a lot of these matches are going to be resolved by having that direct communication with the person who submitted the other match. And really figuring out exactly what does that presentation look like for that other patient, because there can be a lot of variability in saying that someone has developmental delay, for example. Like there's quite a spectrum in terms of what that could look like. There could be other clinical findings like congenital birth defects and things like that that may not be super apparent when you are doing that matchmaking from the outset. And so that communication piece after that initial match is made is really, really important for figuring out whether something is a genuine match and worth following up on.
So, Matt, with that, is it pretty easy within these tools to find, like, if I'm a genetic counselor going in, and let's say I'm in pediatrics, because that's probably within, the scope, like the most common use. Can I go in and say, oh, I see that Matt had a patient with this and know that it's you and reach out to you? How does that part work? Is that pretty obvious in terms of who the either researchers, clinicians associated with that case are? Or do you have to do a little digging outside of the tools?
So currently, the way that the Matchmaker Exchange works is they have some information about who submitted the match that you're matching with, and so most of the time you'll have an email address that you can use to either contact that person directly, and then for some of the databases that are connected to the MME, they have a more sort of centralized system for reaching out. So, for example, the Decipher database that we mentioned earlier on in the program, they have a centralized system where you can basically send a request to the folks who manage Decipher and say, I matched with someone in your database. I'm really interested to learn more about that patient. Would you be able to put me in contact with that clinician? So yeah, I think it kind of varies depending on who you match with. But yeah, generally there is a way to follow up on those matches. It's not just you find out that there's some patient out there in the world that might be similar to yours and there's no way to follow up. Generally, there are ways that you can contact that person.
Yeah, that is such a useful tool because there's been cases that I've had as a genetic counselor where it's just like you're trying to find someone. And when it's really rare, that can be hard to do, even in the age of the internet with this. Nara, what are your thoughts?
Now, I just wanted to add to the question about creating the tools that we are actually using in the background, that that has been a long way since we started with developing the first API from scratch. But around the same time, when we were creating Matchmaker Exchange, the GA4GH, the Global Alliance for Health for Global Health, was also established. And these projects continue working together in the last 15 years. And or as part of the MME or as part of a GA4GH work stream, more or less all the involved pieces have met at least once every 2 weeks in the last 15 years to be exactly discussing what's the best way to develop these tools that are being used in the background. I told you we developed the Matchmaker Exchange API from scratch, but for example, for the variant level matching project, we are now using a standard from J4GH that is the Beacon version 2. And they work to establish these standards that are going to be used to define what is a phenotype, what is a feature, what is a phenotypic feature, what is the gene name, what is the variant nomenclature that's going to be used. That's ongoing work. That is not completely finished. And as you said, the methods that are being used also drive that and drive the changes that are always happening in that process. So it's not a well finished work that we did and we don't need to do anything anymore. It's actually a work that has been ongoing and it's still in development in the last 15 years.
Yeah, that makes me think about just as more and more people adopt this globally, not just within Canada and the United States, but really having this spread out and the adoption really picking up. How does that change the power of these matchmaking tools? Because I'm also thinking about a topic that we've explored on the Phenotype Speaker series. We had Dr. Stephen Kingsmore on, and I'm, blanking on exactly what the episode number was there, but talking about newborn sequencing and just how so many countries are really starting to pilot these programs. I would say the UK is probably the leader in this, but the US has really recently announced the Beacons project. And so with so much more genomic sequencing happening, we're having so much more data and the need to then have more rare diseases diagnosed because they're going to be coming up much more commonly. So I'm just thinking, like, how does this work in changing and having the tools grow, but like the power of these tools must just be amplifying. Matt, am I on the right track with this?
I would say for sure. Like when I think about the Matchmaker Exchange network and matchmaking as as a tool that we use growing, it is definitely for the benefit overall, although it is, I'd say, a double-edged sword. Like obviously having more people connected to one another through matchmaking and more people submitting data to the Matchmaker Exchange network is a great thing in that it increases the likelihood that you will be able to match with someone who is potentially interesting. We generally say within our Care for Her program, about 18% of the time that we put someone into matchmaking, we find another patient out there and it's interesting enough to start those international collaborations. So it's really been a game changer for us in trying to resolve some of these families who have been undiagnosed for, in some cases, many years. With that said, I do think, and this is another thing that we've found in our experiences with matchmaking, is there is the cost in terms of the time that it takes to resolve all of these matches. As I mentioned a bit earlier on, it's not all computational, right? There's a human element to it in terms of figuring out whether you truly have a match with people just because they have the same gene of interest as you do and maybe some broad overlapping clinical features. And so I think as these networks continue to grow, or we turn to more sophisticated ways of matchmaking, like the variant level matching network that Nara mentioned, where instead of having a candidate flagged for a particular patient, you're actually scanning through the sequencing data directly to find those variants of interest. As you move to these more sophisticated forms, we have to think of ways to improve the efficiency of this kind of matchmaking. I think it is very time consuming. As someone who does it for a rare disease program, it is a big part of what I do on a regular basis, reaching out to people, resolving these matches, following up on the collaborations that result from that. And, so we need to think about it almost from an accessibility standpoint. As you mentioned, there are clinicians and genetic counselors who may think about getting involved in it, but realistically, for a lot of these people, they're going to be doing it off the side of their desk. And if we increase the potential for new matches, but they don't have as many tools to help resolve those matches, then that may become almost a barrier as it becomes more popular.
Yeah, that's a really, really astute point. And my lovely producers on the back end, I don't know if it's Joel or Michael, said that it's episode 12, The Future of Pediatric Genetics, that I was referring to with Dr. Stephen Kingsport. You guys are amazing. Orion, I feel like you have something to add here.
I think I'm just, hearing what Matt is talking about and feeling like I think the power of these systems grows exponentially with the number of sort of examples or patients that we're matching on through it. And I think that's, yes, this double-edged sword of both being incredibly amazing in terms of the ability to find matches, but then puts more strain on the work of kind of evaluating them. And I'm really curious what your thoughts are for how that side can scale. What, in terms of and maybe part of this is around VLM. And so would love to hear about that from both of you, but also about kind of, are there ways that we can help computers to help, lighten that load as well? Because ultimately, the more matches, the more good matches that we're generating, the better it is for everyone. And when we started the Matchmaker Exchange in the early days, and we had that special issue that came out, got to work with Peter Krawitz and Peter Robinson on a paper about kind of the birthday paradox, being that, you're 365 days in the year, but basically once you have a classroom of like 23 people, you're more likely than not to have two that have the same birthday, which is usually a smaller number than people you would anticipate. And I think that speaks to the power of these systems. The more, just as the size of these databases grows even a little bit, the matchmaking power grows exponentially, and that ultimately then comes down on the humans who are doing a lot of the work to evaluate. And so,
Yeah. Nara, what do you have to add?
Now, I always kind of push back on that a little bit because I think one of the advantages of the network is that you choose what you're looking for. So, yes, you may get a lot of matches, but that means that what you chose, it's probably not the best candidate. So it will force you to think better of what to include in the network the next time. So in Gene Matcher, for example, we end up putting a cap of 10 genes per individual. You cannot submit more than 10 genes as candidates per individual, because if you do, it means that you didn't think hard enough to really narrow down the best candidates. And then you are going to get a ton of matches if you put 10 genes that you didn't think as you could about them. And you have many genes in there among your 10, probably 9, that are not good candidates, and you're going to get matches that are not specific and not good for your case because you didn't do a little bit more of homework to put the best candidate gene in there. So I think that is one thing. The other thing is on the variant level matching, we kind of lose this control a little bit because I have my data in there and I didn't necessarily choose any variant for my patient. I'm actually letting you look at most of the variants for my patient. And you say I'm interested in variant X, and I will receive the notification about it. But I didn't choose that variant to begin with. So I'm getting a notification about a match that was not my choice and that may not be a good one. So on the variant level matching, you kind of lose this control a little bit. And that's when I think we have to be a little bit more careful on setting limits so people are not being bombarded with matches that they didn't even choose to have to begin with. But I'm wondering, and I have been thinking a lot about how, and everybody's talking about AI nowadays, and we cannot not talk about AI helping us or thinking of AI helping us to be more specific to these matches. And I think that's something, or I was just asking, how can we maybe improve that I'm hoping somehow AI will allow us to be a little bit more specific about the matches, so we can get less of that, because I'm more worried about that on the variant level matching side of things than I am on the gene matching, because the gene matching, both people have to think hard and pick these good candidates for their cases. And then, yes, they may match by chance, but at least you know that two people are thinking harder about it. On the variant level matching is a one-sided, just one person thought of it. The other one was just sitting there passively, got the match, and maybe it's not the right one and they didn't choose to get that match to begin with.
Yeah, Matt.
Makes sense?
Yeah, I agree with you, Nara. Like I think the potential for having a very large number of matches, many of which could be false positives, is higher in variant level matchmaking, where effectively, as mentioned, you're looking through the sequencing data itself rather than a curated one or two genes that you're interested in. And this potential for a number of false positives is something that CareFor has actually looked into on a sort of smaller scale. We built sort of a pilot platform called the one-sided matching platform, or OSMP, where we were basically looking at this within our internal data set. Like if we try and do this variant level matchmaking approach, just within our own data, how many potential matches do you get when you put a given gene into that system? And more importantly, how can we implement some systems or workflows to help reduce the number of potential false positives before you get to a smaller list that you would then reach out to directly. And I think, the principles are somewhat similar to the matchmaker exchange in that having phenotype information available is obviously important, even though, the standardized HPO ontologies won't capture everything perfectly 100%. It's a really great starting point to be able to remove those who are clearly not a good fit for your own patient's presentation. I think some of the other things that are worth considering when you're looking at a large list of potential matches and related to the variants are information about the zygosity of the variant and potentially the inheritance patterns. So if your patient has a homozygous variant, then you're primarily going to be interested in other individuals who also have homozygous versus heterozygous variants. Other things, including the prevalence of those variants in population databases like Nomad, is also really important. The lens that we've sort of been looking at this has been the rare disease space. And so looking for things that are very rare in the general population is also another way to help narrow things down. So I think, as this variant level matching network is being developed, we're thinking sort of critically about like what pieces of information are going to be needed to help reduce that burden on the users at the end.
And we've started talking about it a little bit. I'd like to dive into more of the future and what we think is really going to be impacting this, what we need to be focused on to prepare properly. I was thinking about this, and then I saw that someone also submitted this question. So I figured this is a good transition into some of the audience questions. So if you are watching this live, feel free to put your question in the Q&A box, and hopefully we'll have time to get to it. So yeah, I mean, would love to hear from all three of you of what your thoughts are on the future of MME, and specifically, are there plans to make MME data more easily and publicly accessible, or to enable it to be indexed and discoverable through search engines like Google? This person goes on to say, I recognize that the more closed model was instrumental in getting MME off the ground in its early days, but I'm wondering whether the field has evolved to a point after 10, 15 years where stakeholders would now be comfortable with more open sharing. Thank you for this wonderful talk. Nara, you have definitely a strong answer for this. I see it on your face.
No, it's actually getting more complicated to share data. It's not getting easier to share data. And that's why we started the federated model to begin with. So each institution, each database could keep their governance on the data. And it's not, I think we went a longer way about people changing their minds about sharing the data in terms of like at the beginning, when we started these projects, people didn't want to put that data in any database because they were afraid that they would be scooped. I don't think people worry about that so much nowadays. But on the other hand, the countries are more protective of their own data. It's not the at the beginning it was we needed to worry about the researcher and convince the researcher to share the data. Now we are dealing with an increasingly regulated field that and the regulation is not coming from the researcher or from the institution itself, it's coming from the countries. As the network grows, we want to include databases and data from all around the world. And to work with all around the world, we have to work with regulations that are very different in UK, in US, in Emirates, in Brazil. So it's not as easy to have this data to be queryable by Google. That's not happening. I'm sorry. That is sensitive data, that is individual genomic data that can be used in many different ways. And that's not going to be openly available for query, like in that sense. We are getting more people to join the network, but we have to increase with more and more regulations every day. And in the future, it's, I think, what we have been working now that we are we are adding to the matchmaker exchange, the variant level matching. So now, instead of matching on two genes that were curated by two sides, we are allowing people to query one variant that they have interest on in this bigger set of variants per individual that we have in these databases that have VCF level data, like we call it. So that is what we are working now as a future, and bringing not only research data, but as I said, clinical data, hospital data, patient data into the network. But that will actually require even more regulation because now it's not a gene name that you're putting in the database. It's the 300, depending on how you are filtering the file to begin with, variants per person in the database. And that is much more sensitive than the name of a gene. So
Yeah, complex privacy concerns. Yeah, there's this is complex. Orion, what do you think?
I guess I have a question for Matt and Nara, which is that like if given kind of the global geopolitical environment and increasing regulations on then the healthcare practitioners and researchers putting data in, there's a need to make the matching stronger, have more data applied for the matching. Does that mean that the future of the MME or part of it is building on kind of the MyGene2 more public and gene matcher to some extent, the more public way of sharing that it becomes then something that we're, enabling the public and patients and people with conditions to share their own data, and that then the role is building and managing these systems and helping to educate them on how to use it, or at least have them be the ones who are contributing their own information as a way of bridging that increasing regulatory I guess hurdle, for the sake of having the data to actually be like helping them to find matches.
Matt, I'll let you start. Don't want to dominate. You go.
Yeah, yeah. So yeah, your question, Orion, is around um, whether there should be like greater efforts on the network side to help facilitate
Like
improving the accessibility.
to
Yeah.
or to some extent is because on one hand we want the regulation is increasing, making it harder to share data, but we want even more and more data and more sensitive data to be used for matchmaking to make the algorithms more capable and
Hmm.
lighten the manual work. To me, one possible solution is leaning heavily on the like direct engagement with the patient or public, so it's them being the data contributor at the end of the day. I was just curious what the two of you thought about that.
Yeah, I think there have been, some efforts within the Matchmaker Exchange to facilitate those kinds of connections. There are several nodes. You mentioned, MindGene2 as one of them, Patient Match or another within the Matchmaker Exchange that allow that sort of direct contribution of data. And so yeah, like I think from the level of matching on a gene level side, like there are efforts to improve that accessibility, even in light of increasing restrictions on contributing and sharing this data across international borders. I think that for the variant level matchmaking, it's continuously a, a bit of a tug of war in terms of trying to figure out, um, you know, trying to make the requirements for joining the network as broad and accessible as possible, so that other databases can join, while still retaining that utility that comes from matchmaking with increased access to more sort of individual level information. Nar, do you want to add anything to that?
No, I'll just say, like on the Matchmaker Exchange, there are three nodes that allow direct patient participation. That is, like you said, MyGene2 and Patient Matcher. And Gene Matcher also allow the patient directly to put, but the patient is putting
Right.
name of a gene. That's much easier. On the variant level matching, for a patient to participate directly, they have to find a database that allows them to put their VCF file in there. And as far as I know, the only one that allows that to happen is MyGene2, Gene2MP. Matt, correct me if I'm wrong. But you have to have a system that's well designed for them to put their data in there. The other databases, Orion, are all mostly research databases or clinical laboratories we're trying to bring in now. And there are some patient-led database that we're trying to bring in also, that we're working with to try to get into the network. But they are in the process of developing their own database, and they are on the process of going through IRB requirements and what will be the right consenting for that patient to put that data there and then share the data in the network. So it is a way, maybe, but it is a way that will take a lot of work also to happen. All right, it's not a easy thing for a patient to get. I have my VCF file. Where do I go now to participate on the network? They have to actually work with a database, a system that's already ready to accept and share with right IRB and consenting process. So that's one way to do it. But I don't see the regulations as a problem. I think the regulations are there to help us to keep the data safe and the individuals that are participating in the network safe. And they are needed and they are evolving because everything is evolving and everything that we are generating is evolving. And we are learning and we need to have these security um, systems in place. It's just that what I'm saying, I was just answering that question about how are we going to just make the whole data queryable? That's not going to happen. And each one of these databases, it's actually harbored in one institution that goes by a lot of regulations, and the regulations are from the institution, from the country, from global regulations, and this data is not just going to be all out there. As we already have it all as out there as we can. That is NOMAD, right? NOMAD
Yeah.
has the data from 700,000 genomes and exomes that have been sequenced, but to be all out there, we have to lose at some level some information. So we don't have phenotype information, we don't have contact information, we don't have a way to access what's the phenotype of the individual that has that variant. We don't know where the variant comes from and what phenotype or not was associated with it.
Yeah.
No way to
It's
actually
like we need
parse it.
these guardrails and they're important, but they do have some consequences. But we can't throw out the guardrails just because there are some consequences. And there's another question in the chat that I think is similar to what we've been talking about. In case people don't want their names publicly shared, I just say initials on here. So CG says, hello, all panelists, you're great. I want to ask if matchmaking efforts are enough as they are now, or should we also be pushing for more efforts on sequencing projects for rare diseases and populations from underrepresented countries and regions to better interpret variants and discover through matchmaking efforts also new disease associated genes? So any thoughts on just expanding this more so that our databases are richer and more representative of humans in general, Matt?
I mean, 100%. Matchmaking is only going to be as useful as the data that gets put into it. And I think it's very well known in the genetics field and rare disease fields. I'd even say amongst healthcare fields in general that there are lots of minority populations whose data is underrepresented. And when you have data like that that's underrepresented, it means that on the clinical side, it becomes much more difficult to interpret, especially for rare disease patients. I talked about, knowing the rarity of a genetic change is critical to knowing whether it's interesting or not. If you have a lot fewer of those individuals in your general population databases, it becomes a lot harder to interpret whether those variants are truly rare or you just don't have enough data to give you that answer. And so I definitely think that improvements to our matchmaking networks and protocols have to go hand in hand with improving data generation. And that should especially include a focus on increased sequencing and increased testing availabilities for those from underrepresented communities.
So I just have one more thing to add is as we think of sequencing more, I do think we need more effort in sharing what's already sequenced more. Just imagine how much sequencing has been done in the clinical setting in underrepresented countries right now, right? Just imagine how much sequencing has been done in Brazil, for example, in clinical setting, but that data is not shared. There is more sequencing that needs to be done in countries like Brazil. Not most of the population has been sequenced, and even the population that needs sequencing is not easily accessible for them. So that needs to be done. And many of these countries are working on it. Like Brazil has a large Genomas Brazil project going on to sequence a large sample of the population. But just imagine that in Africa, for example, in the countries in Africa right now, a lot of sequencing has been done, but that sequencing is not being shared. And the same thing in the Emirates, a lot of sequencing has been done, but it's not being shared. So, in terms of sampling, population sampling, I think we have a ton of sequencing that we are not sharing that could be better used for us to understand minor allele frequency of common variants. Not to say that more sequencing doesn't need to be done, but I do think that a lot more data sharing needs to be done. And the question was, is what we have enough? I think we have systems that are very good and that need to be continue work to get better, but I need more people participating in the network. We don't have everybody that needs to participate in the network in there yet. And if we had, it would be much better than what we have now.
Yeah, so I think my question, which is similar to SB's questions in the Q&A box right now, along with that is there are so many different databases and projects. And as you mentioned, different countries. I think of Dr. Bosco, who's really been a leader in rare disease genetic testing in Brazil, genetic testing in general. He's the one that really was key in starting the Brazil rare disease project network that you're referring to. And so, how do we get people to on board so that they are part of this and they can upload this data and share it going along with the regulations, which probably, unfortunately, will be my last question in terms of how much time we have. Anyone have thoughts? And just in terms of, I don't want to use the word recruitment, but basically just having more people and institutions be able to submit this data so we are growing it in an ethical way to go with regulations. Or maybe that's too hard of a question because there isn't an answer, Nara.
No, I think the first thing is we have to show the value, right? And we have to adapt the tools to the needs of different institutions, databases, and countries. That's why we have been trying to be as flexible as possible to bring people to share what they can. We cannot say you can only participate if you do this, and that, because not everybody can follow these rules. It's not because they don't want. They can't do anything if the country say you can't share that one piece of information, period. So I think that's what we have been doing here is trying to make the network accessible by being flexible on how to join and how to participate and what to share. But also we have to show the value of it. And that's what we try to do in conversations like here today, is try to show that it actually can help not only gene discovery, but variant classification. That's a very big problem right now. All these variants of uncertain significance out there. But we also have more than 3,000 rare diseases that we don't know the gene that causes them. And we have 15,000 genes in our genome that we have no idea what they do if there is a pathogenic variant in one of them. So we have a lot of work to do, and sharing data can help us to do it faster.
Yeah.
Yeah,
I agree with that. Matt, final thought.
Yeah, just final thought. I agree with both points that Nara mentioned. It's about keeping the network accessible to those who might be interested in contributing. And I think it's a really important point to talk about utility. I think in terms of getting more widespread uptake, we do need to convince people that this is a really important part of not just rare disease research, but that it could be a really important part for resolving these from a clinical laboratory perspective as well. And so my last thing is there are lots of cohorts, a lot of case reports that mention matchmaking, but if there are other research groups or larger groups out there who are listening to this and haven't collected or published on the utility of matchmaking for their cohort, I'd highly recommend doing it. Because when Care for Rare published our paper a couple of years ago, there were very, very few groups who commented on it. So I think the more groups that comment on it, the better.
Yeah, definitely. Orion.
I was just going to leave with a bit of a call to action for anyone who is listening in terms of, if you have a case or a data set, there is information on matchmakerexchange.org for getting involved in that activity or connecting to that network or using those databases and navigating which one might be the best to work with for it. And then for the variant level matchmaking efforts that Matt and Nara are part of, that's within the Global Alliance for Genomics and Health working group, and
there is a GA4GH webpage also for the event level matching project with an email there and anybody can feel free to contact me by my email, and I think.
Yeah, and we'll include lots of links in the podcast version of this, which will be coming out shortly. And for everyone that tuned in live, thank you so much. It really is such an exciting aspect of the Phenotyp Speaker Series that we get to do this live and actually have people commenting and asking questions and really pushing the needle with these conversations. You're going to see a feedback link in your browser when this webinar ends momentarily, and we're also going to email it to you within the next day because you might be hopping on to your next patient or meeting or whatever you have going on. But we really appreciate if you could take a moment to offer feedback because it helps us improve what we're doing with the series and the topics that we cover. And please head over to phenotyps.com for all types of information, resources, and to learn about so much of what we do at Phenotyps. And if you want to access other episodes, you can go to the resources tab. The speaker series is going to pop up on that drop down menu. All of our installments are on there, as well as podcast players like Spotify, Apple, everything there. And excited to share that our next webinar is going to be next month in March. We had such a popular installment previously talking about proving profitability in genomics that there was such a desire to keep that conversation going and build upon it. So we're going to be talking about building business cases in genomics. So thank you everybody for tuning in. We have had just such a robust conversation here. Nara, Matt, Orion, thank you so much. You provide so much insight here. I have learned a ton. I think I just need to digest all of this now. So thank you so much, everybody. Have a great rest of your day or evenings. Bye.